Gene editing has been performed by farmers since around 8000 BC through selective breeding for superior traits. This, however, is a slow process, and even with modern knowledge, producing a new crop variety takes about 15 years. Through new technologies, we can increase the speed several times over and even go beyond limitations of selective breeding to introduce new traits into crops. Following the discovery of double-stranded DNA and restriction enzymes in the mid-1900s, several techniques emerged to alter DNA strands. At first, genes were added to the cell with methods that gave random results and were very limited in use. Then, applications of gene editing were greatly expanded upon with the discovery of ZFNs in 1985, TALENs in 2011, and CRISPR in 2012. All three genetic editing techniques perform the same task, but ZFNs and TALENs are significantly less efficient and much more labour-intensive. CRISPR can target multiple genes simultaneously, decreasing time spent, and can also be delivered to the target cell through multiple different methods, which, among other advantages, makes it ideal.
The most common tool within the CRISPR toolset is the CRISPR-Cas9. The Cas9 enzyme cuts the DNA at a specific section in the sequence, guided by sgRNA, and modification occurs with either insertion of a new section of DNA, guided into position by a nucleic acid template, or self-repair occurs, where a DNA mutation may arise. This technique also facilitates removal of unwanted sections of DNA, otherwise known as deletions. Utilizing these tools provides geneticists with the ability to target single or multiple alleles. Alleles are expressed by a plant as traits and can potentially be targeted to affect agronomic outcomes such as greater resistance to pathogens, including leaf rust, resilience in varying climatic and soil conditions, such as increasing drought and salinity tolerance, all to ultimately increase crop yields. Phenotypic traits similarly can benefit from gene editing with increasing food and feed nutrient quality, as well as physical and sensory traits, from coloured cotton to sweeter Brussels sprouts. Applications of gene editing do have potential interactions at various stages of development and use, from genomic errors from DNA double-strand breaks, or epistasis, from which an unknown interaction between a gene with another may lead to unintended gene expression. How a microorganism interacts and populates the environment, from a potential change in plant associations or soil chemical gradients. How the exchange of genetic material or gene flow with other relatives will affect weed populations and may influence the ease of controlling them. And importantly, supply chain stakeholders.
Gene-edited crops play a significant role in modern-day farming. For farmers, crop disease can result in significant financial losses due to reduced yields and the need for expensive pesticides or fungicides. Gene-edited crops that are resistant to pests and diseases bring several advantages, such as reduced crop losses, less labour and time invested. For example, farmers growing gene-edited wheat or rice with built-in disease resistance won't need to worry as much about common pathogens, leading to more reliable yields and less crop failure. In extreme weather events and poor soil conditions like high salinity or drought, gene-edited plants with enhanced tolerance to abiotic stresses are valuable in the following ways: expanding growing areas, high yields under stresses, and more reliable planting. Gene editing can make crops more efficient in using nutrients, water, and light, leading to better yields with fewer inputs. For farmers, this translates into higher productivity, lower input needs, and better crop quality.
For consumers, gene editing technology offers numerous advantages. It can reduce the need for pesticides and fertilizers, thereby lowering the cost of food production. Additionally, it could make crops thrive in remote or nutrition-poor soils, improve the nutritional content of food, and enhance product appearance and taste, making them more appealing. However, many consumers remain skeptical of genetically modified food. This is largely due to their reliance on information from social media and word of mouth, much of which is unreliable. Such information is often biased and lacks proper scientific backing. To fully unlock the potential of gene-edited crops, it is essential to enhance public education and ensure transparent communication. This will help build trust and promote broader acceptance, allowing gene editing to contribute more effectively to global food security and nutritional improvement.
The benefits to gene-edited crops for the researcher means increased efficiency, as they will no longer need to screen large numbers of plants for genetic improvement, and instead can target specific parts of the genome to achieve desired breeding goals. This will also mean that researchers will no longer need to wait for a given crop to mature. Instead, new genotypes can be added to the line of crop early in its development and with its desired phenotype displayed at its desired intensity without the need for many generations. Furthermore, this will change how the funding pool is managed. Funds could be redirected to different areas of research, thus increasing productivity over a large range of products. This allows researchers to try innovative ideas in quick succession to test the viability of many traits quickly. Thus, the feasibility of projects can be tested before significant time, and funds are spent on projects that may not be feasible.
Besides increasing yields, gene editing technologies also have the possibility to prolong the shelf life of perishable items. Enhanced shelf life would allow supermarkets to further enhance their ability to supply perishable products such as fruit and vegetables year-round, despite producers being limited in yield by climate, pests and diseases. In terms of a global influence, genetically modified food products can be grown in a country where the climate is suitable and reliable, and allow it to be exported to countries where previously these products may not have been able to reach, such as mangoes to buyers overseas. Consequently, this will increase the demand on transportation services such as the shipping and trucking sectors, and therefore will require more efficient supply chains, while also providing greater job opportunities to those with the relevant skills of business management, logistics and transport roles such as truck driving. Conversely, the already strained supply systems in Australia and globally already struggle to meet demand for truck drivers, leading to the potential for shortages in the short to medium term.
Traditional breeding is slow, but modern technology, especially CRISPR, has sped up the process. CRISPR is now the leading method used to improve traits, reduce weeds, manage stress, increase yield, and extend food shelf life. In the short term, there might be some strain on the supply chain, but the long-term benefit will outweigh the challenges.